Okay, let's dive right into this. Welcome to this explainer, where we're going to look directly under the hood of modern genetics to decode what makes you, well, you. We're going to move way past the basics today and explore the fascinating and, honestly, sometimes mysterious world of complex traits. So get ready, because we are going on a journey from macro population science all the way down to your personal health risk.
Here's our roadmap for today. We'll start by looking beyond simple genetics, unpack what heritability actually means, and hunt for genetic clues. Then we'll tackle the missing heritability mystery, and finally, make sense of what all this means for your actual personal risk.
So, Section 1, Beyond Simple Genetics, moving past the Punnett square. You see, the genetics we all learned in grade school, where one gene neatly equals one trait, that really only tells a tiny fraction of the story. And this brilliantly illustrates the difference. On one side, we have Mendelian traits. These are caused by rare genetic variants that have a massive effect on your biology. Something like hypertrophic cardiomyopathy. They follow those super predictable dominant or recessive patterns we learned about. But on the other side, we have common complex traits. And these are the traits most of us care about every day. They're caused by a combination of tons of common variants that each have a really low effect size on their own. Plus, they are heavily influenced by your environment. You might see an increased risk among siblings, sure, but there is just no simple, clean inheritance pattern here.
And the architecture of these traits gets incredibly fascinating. For instance, we have pleiotropic genes. That just means a single gene can actually affect multiple, completely different traits. There's also the field of pharmacogenomics, which reveals how your unique genetic makeup literally determines how your body reacts to specific drugs. And understanding all this gives us highly actionable insights. A really great example of this is Francis Collins, the former head of the U.S. National Institutes of Health. He actually used his own genetic report, which showed an elevated risk for type 2 diabetes based on his common complex variants, as the ultimate motivation to lose weight and completely revamp his diet and exercise routine. Understanding your complex traits isn't a life sentence, right? It's empowering.
All right, let's move to Section 2 and see how this builds, unpacking heritability and what the numbers actually mean. To really understand this, we need to look at what the numbers actually mean when we talk about a trait being genetic. The formal definition of heritability is the percentage of phenotypic variability, which just means the physical differences we can see in a population that can be explained by genotypic variability, or genetic differences. Notice the key words there, variability and population. I want to emphasize this heavily because it is hands down one of the most misunderstood concepts in science. Heritability absolutely cannot be applied to a single individual. If you hear that schizophrenia or height is 80% heritable, that absolutely does not mean that genetics explains 80% of your specific trait, or that there's an 80% chance you're going to get a disease. It strictly measures the variation within a specific group. Now, what's really interesting about this slide is how heritability is a population-specific metric. Just look at the estimated heritability of height. In European populations, it's estimated at 80%, but in Asian populations, it's estimated at 60%. So did the fundamental biology of human height change? No. What changes is the amount of genetic and environmental variation within those specific groups. Heritability can, and absolutely does, differ between populations. Which brings us to Section 3, hunting for genetic clues. Let's look inside the Genome-Wide Association Study, or GWAS. So, if complex traits are caused by tons of tiny variants, how do we actually find them? Well, think of a Genome-Wide Association Study, a GWAS, as a massive population-level treasure hunt. To run one, scientists follow a strict set of steps. First, they gather an affected cohort, so people with the disease, and an unaffected control cohort from a similar population. Second, they want to maximize the overall diversity of both cohorts so they can observe a really wide range of variants. But, and this is a huge but, step 3, they must minimize the differences in diversity between the two cohorts. If the affected group has a totally different ancestral background than the control group, scientists are going to end up identifying false positives that are just related to ancestry, not the disease itself. Finally, step 4, they scan the entire genome to unbiasedly identify common genetic variations, called SNPs, that are associated with the condition. When they get the results, they plot them on what's incredibly aptly named a Manhattan plot because, well, the data points look like a city skyline. The higher the peak, the more significant the finding. In one major study of schizophrenia, researchers found 108 significant regions in the genome. On chromosome 6 alone, there were 4 distinct peaks.
And the absolute highest skyline peak on this chart corresponds to the MHC locus, making it the most significant finding in the entire study.
But we have to read these plots correctly. A GWAS identifies SNPs that are linked to causative variants. They're basically the signposts, not necessarily the actual causes. Also, that incredibly tall peak at the MHC locus we just looked at, that just means it has the highest statistical significance, meaning it is the least likely to be a false positive. It does not necessarily mean it has the highest odds ratio of actually causing the disease. And remember, because these are complex diseases, an affected individual will likely only have a tiny subset of all the SNPs identified in the study, definitely not all of them. Genetics is very much a game of probabilities.
Let's dive into Section 4, the missing heritability mystery. Where did the rest of the genetics go?
You see, even after doing these massive, incredibly advanced GWAS treasure hunts, we kind of hit a wall. We define missing heritability as the amount of phenotypic variation in a population that we know is caused by genetics, but that we simply cannot attribute to any of our known variants yet. It is literally the ultimate biological mystery that modern science is racing to solve today.
If we look at our total heritability pie here, one slice represents the known variants, the single nucleotide changes we can easily map. But look at that large missing slice. That is the genetic influence we can measure in a population, but can't yet pin down to specific letters in the DNA code.
So, where did it go? Well, scientists suspect a few main culprits are hiding in our DNA. We're likely missing unidentified rare variants that are just too subtle for our current GWS thresholds to pick up. We've got complex gene-environment interactions. We have genes interacting with other genes in ways we haven't mapped yet. And heavily, we suspect uncharacterized structural variations are playing a really major role here. So the crucial point is how this actually plays out in real biology. Let's look back at schizophrenia for a second. Researchers dug into that MHC locus we saw earlier and found a structural variation, specifically a duplication of the C4 gene. This duplication increases RNA levels, which translates to a protein that promotes synaptic pruning during teenage brain development. Well, too much of this protein means too much pruning of neural connections, which physically elevates the risk of developing schizophrenia. It's an absolutely incredible link from a structural gene variant directly to human brain development.
Finally, Section 5, making sense of risk. Let's translate these population stats down to personal health. Now we're going to zoom all the way in. How do we take all these population stats, these towering GWAS peaks and the missing heritability, and actually apply them to an individual reading their own genetic report? Imagine you open your report and read that a specific variant you carry, like that C4 schizophrenia variant, gives you a 25% increase in risk. That sounds terrifying, right? A quarter of your baseline health just seemingly vanished into thin air. But hold on a second, take a breath. That 25%, that is your relative risk increase. To find your actual absolute risk, you have to anchor it to the baseline. And the baseline risk for schizophrenia in the general population is only about 1%. So a 25% relative increase of that 1% only brings your absolute risk to 1.25%. Suddenly, that terrifying 25% jump represents a very modest reality. Context is literally everything. Complex traits demand a complex view. If we look at the total risk architecture for something like schizophrenia, we see it's 80% heritable in the population with an elevated sibling risk. But it is also strongly influenced by the environment, things like in utero malnutrition or certain infections. And if you take a genetic test, you need to be aware that risk predictions actually vary wildly by lab right now. There are no standard guidelines, so different labs select different variants and combine them in completely different ways to guess your risk. And they're entirely leaving out those crucial environmental factors.
Let's apply this context with a little puzzle. Imagine a 45-year-old asymptomatic woman watches a news story on schizophrenia and suddenly worries she might develop it. Based on a general population baseline risk of 1%, what do you think her actual risk is today? Just take a second to think about it.
Her actual risk is less than 1%. We have to apply the concept of conditional risk here. Since most diagnoses occur in teens or young adults, the very fact that she has reached 45 without developing any symptoms means her personal conditional risk has actually dropped below the general population average. Just like heritability, you simply cannot blindly paste a population statistic onto a specific individual without looking at their context. And that leaves us here.
As these GWAS treasure hunts get larger, and as science inevitably hunts down that missing heritability, our genetic reports are going to get much, much more precise. So the question is, when you truly understand the difference between a scary relative percentage and your true absolute risk, how will that empower you to change the way you live your life? Because as we've seen today, your genetics aren't just a static textbook code. They are actionable insights waiting to be decoded. Thank you so much for joining me for this explainer, and I highly encourage you to keep exploring the science of you.